To the organizers of this virtual conference, co-presenters, other guests, friends, ladies and gentlemen, good morning. I am privileged to present to you one of the component studies of my dissertation, which I entitled Utilization of Selected Microalgal Species as Natural Food for Common Carp Fry, Cyprinus carpiolineus. Presenting to you is yours truly from the University of Santo Tomas, Manila and Mariano Marcos State University, City of Batac, Ilocos Norte, Philippines. Microalgae, are commonly known as phytoplankton, are the base or the bottom of the aquatic food chain. They are the earliest and oldest forms in the archipelago. In the aquatic era, phytoplankton has been the source of natural food, pharmaceutical products, and other industrially important ones. Hence, they are being mass-produced and provide it to aquatic organisms. However, limited resources of mass-produced phytoplankton are still under study. Thus, this study was undertaken. Objectives of this study are to evaluate the proximate chemical composition of select microalgal species found in the freshwater resources of Carigara Bay, mass-produce select microalgal species, provide the mass-produced microalgal species as natural food for common carp fry, Cyprinus carpiolineus, and assess the proximate chemical composition of common carp fry, Cyprinus carpiolineus, fed with microalgae as natural food. There are 9 freshwater resources which are the sampling sites of this study. These resources are located at the surrounding coastal towns of Carigara Bay, namely Capoocan, Carigara, Parugo, San Miguel, and Babatngon. Water samples were gathered using plexiglass sampler and brought to the laboratory for isolation and purification. Using culture medium, the Binangonan Research Station Fantastico medium or the BRSP medium, phytoplankton were isolated, purified, and optimized, semi-mass-produced in 500 ml. After that, it was mass-produced into 10-liter containers and finally into 30-liter containers. Figure 1 shows the 4 phytoplankton that were selected after their growth capacity in few hours only and less microbial contamination while mass producing them. These are Asterococcus limneticus, Chlorococcum humicala, Anabaena azaleae, and Oscillatoria limnetica. Moreover, Table 1 presents the proximate chemical composition analysis of fry mass and the 4 selected phytoplankton. The table explains that Chlorococcum humicala had the highest ash content and Oscillatoria limnetica had the highest moisture content. Furthermore, fry mass gave the highest crude fat and protein contents with 7 and 33% components respectively. Though the compositions were not subjected to statistical analysis, Asterococcus limneticus gave the highest crude fat and protein contents numerically compared to the other 3 phytoplankton being studied. Hence, the proximate chemical composition of the 4 phytoplankton confirmed their potential source of nutrients for natural food, pharmaceutical products, and industry-driven components. Figure 2 presents the mean length of common carp fry with different treatments. The mean length of common carp fry in all treatments with time. So the highest growth was obtained in 6 weeks of feeding. Micola showed the highest growth in length. Furthermore, the treatment is more effective as natural food at the 4th week of feeding experiment, where the 2-factor ANOVA in table 2 showed that there are significant differences in the mean length of the common carp fry, indicating now that the fry feeding experiment had a significant effect now on the length of the common carp fry at 4th week of trial. Moreover, figure 3 presents the mean weight of the common carp fry fed with different treatments from the initial week of up to 6 weeks of feeding trial. The figure expounds that the growth in the mean weight of common carp fry in the different treatments increased in time. And the 6 weeks of weaning period, common carp fry fed with fry mash had the highest growth in weight. And to note, the natural food is more efficient in the 5th week and 6 weeks of trial, where the 2-factor ANOVA in table 3 showed that there are significant differences in the mean weight of the fry, indicating now that the type of treatment does have a significant effect on the weight of the common carp fry at the 5th and 6th weeks of trial. The proximate chemical composition analysis of the common carp fry fed with different treatments is shown in this table. Table 4, common carp fry fed with Chlorococcum humicula or treatment 3 had the highest ash content, and the common carp fry fed with Oscillatoria limnetica or treatment 5, which means that the treatments were not subjected to statistical analysis. Treatments were not complete. Furthermore, figure 4 shows the percent survival rates of common fry in 6 weeks of feeding experiment. Fed with the different treatments from treatments 1 to 5 had 90 to 95% weekly survival. Finally, table 5 presents the physicochemical parameters gathered during the 6-week of feeding trial. The ammonia was less than 0.25 ppm, while the dissolved oxygen ranged from 9.81 to 10.0 milligram per liter, from 31 and 30.2 to 31.5 degrees Celsius respectively. Indeed, the freshwater resources of Carigara Bay, located in the 5 upper shoreline municipalities of the province of Leyte, are still understudied in terms of phytoplankton diversity and applications. With the result of the study, freshwater resources are home to numerous species of phytoplankton which can be isolated, identified, and mass-produced to generate natural food necessary to many aquatic organisms in their fry and post-fry stage. The researcher recommends the following purification and culture studies of the other isolated phytoplankton, culture studies of Phylum Bacillar phyta, and ends my presentation. Thank you very much for your attention.